Hello, and welcome to our first lecture in this new series on drugs and human behavior. I want to talk first about brain structure and function. It's of course very important for us to understand how the brain works and functions in order for us to understand how drugs influence human behavior. I want to start briefly by introducing the basic idea of psychopharmacology. Psychopharmacology is a subdivision of pharmacology, which is simply the science of how drugs affect the body. We're particularly interested in how drugs affect the brain and thereby behavior. So we're going to spend a lot of time talking about where in the brain drugs are influencing behavior, how they're influencing behavior, and in general what we can sort of do about that and what's important to understand about that. Some important pieces of understanding this entire puzzle are understanding what we call pharmacokinetics, which is knowledge of how the body handles drugs, so understanding brain anatomy, how drugs end up in the brain, how they influence the brain, and also understanding how the body handles drugs, gets rid of them, that sort of thing. Pharmacodynamics is another important piece of this puzzle in which we look at interactions with the drug and its receptor, or the structure on which the drug interacts to produce its effects. So keep that in mind as we start talking about brain structure and function. We're going to start with looking at the architecture of the overall nervous system, take a look at the subdivisions of the nervous system. We'll go down to the smaller structures and talk about structure and functions of neurons, look at how they undergo a process which we call neurotransmission. Part of that involves neurotransmitters and their receptors, and then finally those systems that come together in which those neurotransmitters and the receptors influence behavior. But let's start by taking a look at the architecture of the nervous system. Our central nervous system is the major subdivision of our nervous system, which includes simply the brain and the spinal cord. Everything else is the peripheral nervous system. In the peripheral nervous system, we have the somatic nervous system and the autonomic nervous system. The somatic nervous system is that nervous system which controls our voluntary muscles and also conveys sensory information back to the central nervous system. So you can think of the somatic nervous system as that nervous system that interacts with our environment. The autonomic nervous system, by contrast, controls our internal environment. And so it contains two different subsystems, the sympathetic nervous system and the parasympathetic nervous system. The sympathetic nervous system is primarily involved in expending energy, so getting our resources rallied when we need to fight or flee. The parasympathetic nervous system is involved in calming us and conserving energy. This will become important as we start looking at how different drugs influence these different nervous systems. So we'll start off by taking a look at how we navigate the nervous system. There are different ways to think about the different directions, or there are different ways we describe directions in the nervous system. These terms are important as we start going into more in-depth readings about the nervous system. So we'll then take a look at the central nervous system, the spinal column and brain, and then talk about the peripheral nervous systems. So the first thing to get out of the way is understanding how we talk about directions in the nervous system. So when somebody talks to you and says this occurs in the dorsal lateral prefrontal cortex, it's important for you to understand what those words mean. So we'll start with what we call the dorsal side of the brain, or the dorsal side of the body. The dorsal side of the brain is the top side of the brain, so the top of your head. The ventral side of your brain is the lower side, or that sort of down towards your mouth and jaw, or the bottom side of your brain. This is very different for your torso and the rest of your body. For your torso, the dorsal side of your torso is your back, and the ventral side is your stomach. The reason for that is these terms were essentially invented to describe animals that walk on all fours. So if you think of how that would look, the top of a cat's head is in the same direction as its back. So that's the dorsal side. And of course, the shark has a dorsal fin, and that's kind of the easiest way to think about that. The ventral side is the front, or your stomach, or the lower part of your brain. Now, when it comes to the brain, the front of your brain is what we call the anterior portion, and the back is what we call the posterior portion. For all body function, or all body directions, we talk about medial and lateral. So medial are things that are towards the center line of the body, so towards the spinal cord, and lateral, those are things that are towards the outside. So when we talk about something like the dorsal lateral prefrontal cortex, we're talking about something that's at the top of the brain towards the outside, that's in the prefrontal area. We'll talk more about what that means here in a minute. Now, we may look at some different brain sections throughout the semester. So a coronal section is one of a slice that goes directly across your skull, sort of like from temple to temple. A sagittal section is one that goes from front to back. And then a cross section is one that would be as if you simply sliced your head sort of right at your eyeballs to the back of your head. And that would be a cross section. So I've provided an entire list of these different directions and their kind of definitions. We have dorsal, toward the back, or the top of the brain. Ventral, toward the stomach or away from the backside or the bottom part of your brain. Anterior is toward the front, posterior is toward the back. Superior is above, inferior is below. Lateral is to the side away from the midline. Medial is toward the midline. Proximal is close. Distal is far. Ipsilateral is on the same side of the body. Contralateral is on the opposite side of the body. And then we have our different planes. Also provided here for your reference, so you can come back to this if you need it. So if you come across one of these terms, you might have them lamina, column, tract. We're mostly going to talk about nerves, which is a set of axons, which form things like the cranial nerves. We'll talk about nucleus. These are clusters of neurons within the central nervous system. Ganglion is a cluster of neural cell bodies, usually outside the central nervous system. We'll talk about gyrus or gyri, which is those outcroppings on the surface of the brain. A sulci is a fold or groove in the brain. And a fissure is a long, sort of deep sulcus. So the central fissure goes straight down the mid part of your brain. So these are simply ways that we often use to talk about getting our way around the brain's anatomy. So I want to spend more time talking about the actual parts of the nervous systems, the things that are really going to be important to understanding its function. The spinal column is, of course, the information superhighway. It takes everything from the brain and moves it out towards the rest of the body, so lots of information coming in and going back out. Obviously, we have to have an intact spinal column to breathe and to feel and to walk and talk and do all the things that we do. So the spinal column extends from the medulla, which is at the very base of the brainstem, all the way down to the sacrum, which is our tailbone. So everything in between on your back is your spinal column. This includes 3 separate sections: the cervical vertebrae, which we kind of think of as our neck bones, thoracic vertebrae, and then finally our lumbar vertebrae, which is that part that kind of curves in when we think about having lumbar support. We have cervical nerves, thoracic nerves, lumbar nerves, and sacral nerves are all part of what we think of as our spinal column. The brain, of course, no surprise, is far more complicated. We're going to talk about the brain's different sections. There are a lot of different ways to sort of slice up the brain, as it were. We'll talk about the hindbrain, the midbrain, and the forebrain, and its subcomponents. So the hindbrain consists primarily of the medulla, the cerebellum, and the pons. The medulla and the pons are part of typically what we call the brain stem, which also includes part of the mesencephalon, which we'll talk about here in a moment, which is often referred to as the midbrain. And then the forebrain includes the thalamus, the hypothalamus, which is the diencephalon, and then the telencephalon includes the cerebral cortex, which is the bulk of the brain, the limbic system, which is involved in memory and emotions, and then the basal ganglia, which are involved in motor coordination. So we'll take these kind of one at a time. So we'll start with the hindbrain. The hindbrain includes most of the brainstem. The brainstem includes the medulla, the pons, and the midbrain. The medulla and the pons are particularly important. The medulla in particular is what keeps us alive. The cerebellum is Latin for small brain. You can see it looks like a smaller version of the brain sticking right off the back of the brainstem, and it has some important functions as well. So let's start with the medulla. The medulla is located just above the spinal cord. It's like an enlarged extension of the spinal cord. This is the part of the brain that's responsible for our vital reflexes, such as breathing, heart rate, vomiting, salivation, coughing, and sneezing, all the things that keep us alive. There's also structures within there that are involved in our perception of pain. And so when we talk about pain perception and how to manage pain with opioid analgesics, we'll be revisiting that very closely. The pons is Latin for bridge. This is just simply where axons from each half of the brain cross to the opposite side of the spinal cord, so that the left hemisphere is controlling the right side of the body, and the right hemisphere, of course, controls the left. So this is just simply a crossing over point. The cerebellum helps regulate our motor movement, balance, coordination, and temporal perception. It's also important for shifting attention between auditory and visual stimuli. This becomes very important when we start talking about the effects of drugs on behavior because the cerebellum plays a pretty significant role. So when we start talking about things like how alcohol affects motor coordination, we're going to be taking a look at the cerebellum. When we talk about why marijuana smoking alters temporal perception, that is why time seems to pass at a different speed, we're again going to be back at the cerebellum. So it's a really important part of what we're going to be talking about. Excuse me. We will be talking about drugs and how they affect various parts of the hindbrain. Some very important components of understanding this piece are how depressant drugs such as alcohol, barbiturates, benzodiazepines, and some opioids exert an effect on the brainstem activating system. In particular, we can get depression in breathing, and in fact, people sometimes will stop breathing as a result of their drug use or abuse. And so it's a really important component for us to think about. Again, alcohol, as I mentioned a moment ago, impairs the cerebellum. This can result in what we call alcohol-induced ataxia, which is just a loss of motor coordination. And so we want to think very carefully about how these components of the hindbrain are affected by drugs, and this will come up in a variety of places. The midbrain, which is often called the mesencephalon, includes the tectum, which is simply the roof of the midbrain. The superior colliculus and inferior colliculus are important because they process sensory information. The superior colliculus, for example, usually contains, in most species, both visual and auditory information, and in humans it contains both visual and auditory information. So we can integrate our sense of space through both of those senses. The tegmentum contains nuclei for the cranial nerves and is part of what we call the reticular formation, and again that's going to become important later. And then finally the substantia nigra, which give rise to the dopamine-containing pathways, facilitating readiness for movement. The substantia nigra is a really important part of our motor system, which integrates with the basal ganglia for us to be able to move. This part of the brain is implicated in Parkinson's disease, and we'll talk about how drugs used to treat schizophrenia can have effects on the substantia nigra and the rest of the basal ganglia. The forebrain is essentially the rest of the brain, which is divided in the diencephalon, which is the thalamus and the hypothalamus, and the telencephalon, which include the basal ganglia, the limbic system, and the cerebral cortex. And we'll just take these one at a time. So the thalamus is our sensory switchboard. It's really all its primary function is a place for which the senses move on in their organization towards the cerebral cortex. So the visual system has a very large set of neurons in what's called the lateral geniculate nucleus, and the auditory system in the medial geniculate nucleus. So these are really important parts of our sensory system. The hypothalamus is something we'll spend a lot more time talking about in this particular course. It has 11 major nuclei. It's involved in the regulation of our autonomic nervous system. So when we talk about things like amphetamines and how they affect our body, one of the things they do is they kick in that sympathetic nervous system, and that's going to occur via things like the hypothalamus. The hypothalamus is involved in releasing hormone-releasing factors, so things like corticotrophin-releasing factor, which are involved in our reproductive hormones, and we will spend some time talking about those. This is also an important site of action for many psychotropic drugs. In fact, the hypothalamus plays an important role in a lot of motivating behaviors and in our sense of reward. And so we'll talk more about its role in drugs throughout this semester. The telencephalon includes 3 different subcomponents: the basal ganglia, the limbic system, and the cortex. The basal ganglia, as I was just mentioning a moment ago, is primarily involved in our motor coordination and our ability to move, walk, talk, do those sorts of things. The limbic system is involved in a variety of functions. Primarily, we're going to talk about its role in memory and emotion and its role in the reward pathway. Finally, then we have the cerebral cortex, which we'll spend some more time talking about here in a moment. Let's start with the basal ganglia. This comprises of the caudate nucleus, the putamen, and the globus pallidus. The globus pallidus is actually a part of the basal ganglia which is sometimes targeted in treatments of Parkinson's disease. There's an intricate way in which these parts all work together to coordinate our motor movements, and when that gets upset, that balance of dopamine-producing neurons gets upset in Parkinson's disease, then we end up with an inability to provide coordinated motor movements. So those are the parts of the basal ganglia. So again, this is associated with planning of motor movement and with aspects of memory and emotional expression. It's also important for attention, language planning, and other cognitive functions. In particular, the basal ganglia seem to be involved in such tasks as implicit or unconscious forms of memory. This is located beneath the cerebral cortex in what we call subcortical areas, again, which include the caudate nucleus, the putamen, and the globus pallidus. The limbic system is the second major subdivision of the telencephalon. Its primary components that we'll talk about are the amygdala and the hippocampus. The amygdala is the part of our brain which is primarily responsible for our very primal emotions. Fear, anger seem to be particularly involved with the amygdala. The hippocampus is the place in which our memories are formed and plays a very significant role in our ability to do things like navigate in spatial areas, so our spatial memory. So the limbic system together contributes to our regulation of mood, affect, emotion, responses to emotional experiences, and is also part of our reward circuit. And we'll talk about the reward circuit here in a couple lectures when we talk about addiction. And so it's going to be a really important part of that process. The final part of the telencephalon is really the most significant part, which is the cerebral cortex. And the cerebral cortex, or what we sometimes call the neocortex, is divided into 4 different lobes, and of course 2 halves. So there's the left half and the right half, and each half is divided into the frontal lobes, the temporal lobe, the parietal lobe, and the occipital lobe. We'll talk about these each individually, but you can see there is some clear specialization to different parts of the brain. So the frontal association areas have a bunch of different specific functions. As we move towards the posterior part of the frontal lobe, we get what's called Broca's area, which is responsible for our speech production, which you can see is located right next to the motor cortex. Then we move to the parietal lobe, we get our somatosensory cortex, which is our senses, body senses, located with our sense of taste, our ability to understand speech, our ability to read. And then we get the temporal lobes, which are involved in our sense of smell and hearing and auditory associations, and also language. And then finally we get the occipital lobe, which is primarily or entirely devoted to vision. So the frontal lobe is involved in tasks such as working memory and executive functioning. So people who have head injuries often have what we call executive function difficulties. Usually that means that they have difficulty planning and coordinating, understanding risk, etc. We'll talk about how different executive functions get disrupted during different drug abuse episodes and through different disease processes. So motor planning and coordination is accomplished by the frontal lobe, and this is also where language production occurs. The temporal lobes, which are located just beneath your temples, have a couple primary functions. This is where our primary auditory cortex is, and there are quite a few language functions involved here as well. The parietal lobe is responsible for visual attention and spatial orienting, particularly the right parietal lobe. The left parietal lobe is often involved in mathematics, reading, other complex symbol-based functions. And then finally, the occipital lobe is located all the way at the back of the cerebral cortex. This is where our primary and secondary visual cortices are. And in fact, the occipital lobe is entirely devoted to our visual system. So we'll spend very little time talking about the occipital lobe in this particular course because very few drugs that we're going to talk about have an influence on our visual system. So that's a quick rundown of our central nervous system. I want to turn next to talk about our peripheral nervous system. This is, of course, the part of the nervous system that is outside of the skull and spine. We have the somatic nervous system. This is a division of the peripheral nervous system that interacts with the environment, conducts sensor information from receptors to the central nervous system, and from the central nervous system to the skeletal muscular system. The autonomic nervous system is the division of the peripheral nervous system that participates in the regulation of the body's internal environment. A subdivision of the autonomic nervous system is the sympathetic nervous system. This is the division that mobilizes energy and resources during stressful situations, something like that fight or flight response that we've talked about in many courses. High stress situation will kick in the sympathetic nervous system that allows us to gather energy and run, engage in lots of very strong activities. So whenever the sympathetic nervous system is engaged, you're going to know that there are some specific physiological responses, and we'll take a look at that here in a minute. The parasympathetic nervous system is generally our sort of body's default state. It's what calms the body, conserves energy during non-stressful times. So most of the time, our parasympathetic nervous system is doing its thing. The sympathetic nervous system is what arouses our energy when we need it. So to give you an idea what happens when we have a sympathetic nervous system response, our pupils will dilate, our bronchi will relax. The reason our bronchi will relax, it allows us to exchange oxygen very quickly, which is of course very important for cellular functioning. The heart will accelerate and strengthen. Our stomach or intestines will inhibit their activity so we won't be hungry. And the blood vessels of internal organs will constrict. So what happens with certain drugs of use and abuse is you will get what's called the sympathetic nervous system response. So amphetamines are a perfect example. Amphetamines engage the sympathetic nervous system. Pupils will dilate, bronchi will relax, heart will strengthen and accelerate. That can become problematic if that's over the long term. You can get scarring, the heart muscle wears out, your stomach and intestines will inhibit. That's the reason why people who take amphetamines don't eat. That's obviously not good over a long run. And probably the most problematic is the blood vessels of internal organs will constrict, which means that those organs are not getting the blood supply that they need. And as a result, you can get pretty significant damage to the liver, to intestines, to the kidneys, etc. So we're going to talk more about that when we reach those issues. The next really important thing for us to talk about are the structure and functions of neurons. And these are, of course, the individual cells of the nervous system. And there are several different types that these we'll talk about. We'll talk about neurons and different kinds of glial cells. Then we'll spend some time talking about the major structures of a neuron, and then get down to the sort of nitty-gritty a little bit to talk about the cellular membrane surrounding a neuron, because it's a really important part of understanding how neurons function, and in particular how drugs affect that functioning. But let's start by talking about neurons and glial cells. So neurons are the basic functional unit of the nervous system. These are the individual cells of the nervous system. Nerves then are bundles of axons of neurons. So neurons are individual cells, and the nerves are bundled up neurons. And then glial cells are the support cells of the nervous system. And these provide important critical functions, and we'll talk about these briefly, and they'll come up in a variety of places. We're going to spend most of our interest, though, talking about neurons, because they're the ones who are actually creating behavior. But that doesn't mean the glial cells don't play important roles. So glial cells are the support cells of the nervous system. And there are different kinds of glial cells. There's what we call an oligodendrocyte. And an oligodendrocyte is particularly important for sensory and motor functioning. So if you look over here, this oligodendrocyte is wrapping around the axon of a neuron, which creates what we call the myelin sheath. And this myelin sheath is really important for speeding up neural processing. Microglia are important for kind of cleaning up waste products of neurons. And then the astrocytes are important for providing nutrients for the neuron. You can see they are attached here to blood vessels, and so they're interacting with the blood system to, again, send off products and also take back in things that the brain needs. So these functions include things like maintaining homeostasis, forming the myelin sheath, and providing support and protection for neurons. So these are the support cells of the nervous system. Unfortunately, these are also often the cells that can end up causing brain tumors. So you may have heard of what's called a glioblastoma. That's a cancerous cell caused by a malfunctioning glial cells. So keep in mind that the brain uses a great deal of blood, oxygen, glucose, etc., and so it has a lot of blood vessels. And as a result, it has a lot of things that can also go wrong with it. So we'll talk a great deal about that as we move through the semester. Excuse me. The major structures of a neuron include the cell body, axons, and dendrites. So we'll take a look at these individually. But you'll see the cell body is exactly what it sounds like. It's the major body of the cell. It contains all of the other things that a cell body is supposed to, the nucleus, mitochondria, etc. The axon is generally a single fiber that moves away from the cell body. Axons can be millimeters long, or they can be meters long. In fact, there are no synapses in your somatic nervous system except for those at the spinal cord. So when you step on a tack on your toe, that nervous impulse of that feeling travels from your toe up to your spinal column, all via single axons. And so those cells actually stretch all the way from your toe to your spinal cord. So they're actually several feet long. So it's important to understand. So if you look at these major parts of a neuron, you can spend some time looking at this in your own time. We're going to spend most of our time talking about these neurotransmitters down here at the bottom. We'll get into some of these cell body parts. Obviously, the nucleus contains the DNA. We have cytoplasm, endoplasmic reticulum, ribosomes, mitochondria. The Golgi complex will be important. These modify and package proteins, and these are oftentimes where some of our neurotransmitters will come from. We're going to spend most of our time talking about dendrites, the axons, the myelin sheath, and the terminal branches of the axon. So this is a basic motor neuron where we have dendrites, be the receiving end of the neuron, the nucleus or the cell body, and the cell body, this axon that travels down to the muscle fiber. So for example, your big toe, this fiber right here will travel from your spinal column all the way down to your toe, and then it will reach its muscles and cause those muscle fibers to fire. In the opposite direction, we have a sensory neuron. We might have, let's say again, we're stepping on a tack or something, causes these pain receptors to fire that travels up this axon up to the spinal column. So looking at these individually, the cell body has, of course, the cell nucleus, which is where the cellular DNA is. We'll talk about the cellular DNA in a variety of places when we start talking about things like epigenetics and pharmacogenetics and pharmacogenomics. The mitochondria is, of course, the cell powerhouse. And the Golgi apparatus modifies and packages proteins. And this, again, can become important when we start talking about how neurotransmitters are packaged, sent down these microtubules down the length of the axon to be released at the end terminal buttons of the axon. So the axons are our next major function. This is the transmitting end of a neuron. Neurotransmitters are released from the terminal branches or terminal buttons. Some people pronounce that boutons. It's buttons to me, so we're going to call them buttons. So those neurotransmitters are released from these terminal branches that then bind to the postsynaptic cell on its dendrite, causing it to generate an action potential, which then causes it to release its neurotransmitters. And these neurotransmitters are what drugs have a tendency to influence. The myelin sheath is an important part of the neuron. It covers the axon of many neurons. This is usually what we call white matter. And it speeds up the process of neural conduction. So myelin is very important for sensory and motor functioning. Multiple sclerosis, for example, is a disease in which the body's immune system attacks the myelin sheath. And when that myelin sheath gets degraded, the motor and sensory functions of patients end up getting degraded. So that myelin sheath is particularly important. Synaptic vesicles, then, are membrane sacs which contain neurotransmitters. These vesicles contain, it's like a little sac full of neurotransmitters. This vesicle will then bind with the cell wall of the terminal branch of the axon. Then when an action potential reaches the terminal branch of an axon, it will cause those neurotransmitters to be released into the synapse. And so those neurotransmitters can then have their effect on the postsynaptic neuron at the dendrites. So the dendrites are the final major structure of a neuron I want to talk about. This is the receiving end of a neuron. This includes what we call dendritic spines, and this is where specialized receptors are for neurotransmitters. Now both the axon and the dendrites have receptors for neurotransmitters. Dendrites have specialized receptors for neurotransmitters that are released from the presynaptic neuron. The axon of a neuron has also receptors for neurotransmitters that usually tell it to take those neurotransmitters back into it, so what we call reuptake, or causes it to stop releasing neurotransmitters. And so we'll talk about these different processes as we move along. The cell membrane is the final important structure, and the cell membrane has some really important properties because this is what allows it to actually generate electricity. The cell membrane is a semipermeable membrane that encloses the neuron, and this is critical to neurotransmission. The process of neurotransmission involves creating an electrical signal that causes the release of neurotransmitters. And so this cell membrane has some properties that allow it to do that. So if we look here, we have lipid molecules in the membrane. Those block any ions from coming in or out. There are ion channels that can be opened and closed. And without getting too far down into the nitty-gritty details of how the action potential is generated, there are different numbers of different kinds of ions both inside and outside the cell. And that creates a pressure gradient, and it creates what we call a resting membrane potential. That is because there is a difference in the charge outside and inside the cell. It's functioning like almost like a battery, in which there is an electrical charge or electrical potential difference. And so these ion channels open and close to change that electrical charge. So there's a resting membrane potential. And so there's constantly a charge on the resting membrane. When an action potential is generated, it does what's called depolarizing. So it depolarizes or creates this electrical charge that travels the length of the axon that causes those vesicles to fuse with the cellular membrane and release the neurotransmitter into the synapse. Those neurotransmitters, these chemicals, then interact with the next neuron, causing it to create this kind of electrical charge. So we'll talk about this whole process of neurotransmission, but this cell membrane is how that... So this process of neurotransmission occurs via neurotransmitters. So this is how neurons communicate, by relaying information by releasing neurotransmitters. This occurs at what's called the synapse. This is a narrow space between the terminal button of one neuron and the dendrite of another. Synapse is just simply a space between the two neurons. So there is no direct connection between the two neurons. It occurs at this level of the synapse. So if we look here, you can see what's called the synaptic cleft, which is the space between the two neurons. That's the synapse. It's just simply a gap, nothing more. The end bulb of the axon contains vesicles, which you can see are fusing to the cell membrane, releasing their neurotransmitters into the synaptic cleft, which are then binding to the dendrite of that receiving neuron on those receptor sites. So this is the whole process. This nervous impulse, or this electrical impulse, arrives here, causes these neurotransmitters to fuse, causing what's called exocytosis. Those then bind with the dendrite, causing it to fire. So we refer to the axon as the presynaptic neuron and the dendrite as the postsynaptic neuron. So these neurotransmitters are chemicals that are released by the presynaptic neuron, and we're going to get into great detail as to what these neurotransmitters are shortly. There are numerous kinds of neurotransmitters, and they will be a critical part of understanding how drugs influence behavior. The important part at this point is to understand what's called the action potential. And the action potential is a neural impulse in which an electrical charge travels down the axon of a neuron, causing the release of neurotransmitters. So what happens is neurotransmitters bind to the dendrite, causing a change in the cellular membrane, which causes these ion channels to open. They're called voltage-gated ion channels. So once that charge reaches a certain level, these start opening up in the length of the axon. And it's essentially a chain reaction which happens very quickly. Travels down the length of the neuron very rapidly. When it reaches the end of the axon, causes the release of those neurotransmitters. So this nervous impulse or this neural impulse is a critical part of that. And all occurs because neurotransmitters bind to the dendrite, change the membrane potential enough to generate an action potential through these voltage-gated ion channels. Ions flood across the membrane, causing this charge that travels very quickly down to the end of the neuron. Important to understand this is an all or none process. That is, an action potential is going to occur or it's not. There's no big action potential, there's no small action potential. It just happens or doesn't. Once there's enough neurotransmitter in the synapse, that is, enough neurotransmitters that are bound to the dendrite, the neuron will fire. This level of neurotransmitter is called the threshold. Once you cross the threshold, an action potential is going to happen. This process is stopped when there is no longer enough neurotransmitter to keep causing an action potential. So it's stopped by reuptake, that is, neurotransmitters are taken back into the presynaptic neuron. Enzyme deactivation, these are enzymes that break down the neurotransmitters in the synapse. And autoreception, this occurs when the neurotransmitter itself binds to the receptor sites on the end of the presynaptic neuron at the axon and tell it to stop releasing neurotransmitters. So it will essentially stop release of neurotransmitters. So all of these are areas in which drugs can have effects on behavior by increasing or decreasing the amount of neurotransmitters, by increasing or decreasing the effectiveness of neurotransmitters in this process. So everything we're going to talk about is tied right down to this. So here's how this happens at the synapse. We have that action potential that's generated. Those neurotransmitters are synthesized from chemical building blocks called precursors. So they're synthesized either in the axon itself or all the way up at the cell body. Those neurotransmitters are stored and packaged in vesicles. Action potentials then cause these vesicles to fuse to the presynaptic membrane and release their contacts into the synapse. Those neurotransmitters then bind to postsynaptic receptors. And again, this process of neurotransmission is stopped by reuptake, where the neurotransmitter is taken back in to the presynaptic neuron through an active process. They are deactivated by enzymes or through autoreception, where the neurotransmitter binds to the presynaptic neuron and says, please stop releasing neurotransmitters. So different types of neurotransmitters include acetylcholine, which is involved in memory and stimulates movement, serotonin, which is involved in mood, sleep, and appetite, GABA, which is gamma-aminobutyric acid, is involved in sleep and inhibits movement, glutamate, also involved in memory formation, norepinephrine, involved in arousal and mood, dopamine, which is involved in control of movement and sensations of pleasure, and endorphins, which are involved in pain relief. Now, there are different subcategories of these, but these are the general categories of neurotransmitters. So we have all of these steps in neurotransmitter action that are, of course, very important that we've already talked about. We're going to take a little bit more in-depth detail at how neurotransmitters are formed and the receptors themselves. So we'll start with synthesis. Synthesis is simply the formation of neurotransmitters. And this occurs through the chemical processes, much like protein formation, that you may have heard about in other biology classes. So we have precursors of the main ingredient, which are brought into the neuron via the bloodstream, taken up by the cell body and or terminal, and then they come from substances in our diet. So for example, some neurotransmitters are actually steroid hormones like testosterone or estrogen. Those are formed from cholesterol and other kinds of fats. So we have different substances in our diet that actually help us form different neurotransmitters. So the enzymes then put these ingredients together to form neurotransmitters. And so this is much like other different biological cycles you may have learned about in other courses. These neurotransmitters are then stored in vesicles. They are done so to concentrate them so they can release many of them at once in order to generate that action potential. Leaking out a neurotransmitter molecule at a time will not cause an action potential, but releasing a bunch of them will. It also protects them, so they kind of get stored and protected, and then they can be released. They're released via what's called exocytosis. Again, this is probably a term that you've heard. Endocytosis is how something is taken into a cell. Exocytosis is how something gets out of a cell. So those vesicles then fuse with the presynaptic membrane and release neurotransmitters into the synapse. So they bind to that membrane and are released. They're fused with that membrane and are released. Binding then is the attachment of a neurotransmitter to a receptor. So when a transmitter attaches to a receptor, we call that binding. This is important because drugs can affect this process. So for example, we're going to talk about a drug called naloxone. And naloxone binds to the opioid receptor so that opioids can't bind to it and thereby block its action. So attaching a transmitter to a receptor is called binding. And again, sometimes binding can cause that receptor to initiate a response. So if an opioid responds or binds to that receptor, it's going to generate an opioid response. Whereas if naloxone binds to that receptor, it's going to do nothing, but it's also going to keep that opioid from being able to take that receptor site, and so it's going to completely make it ineffective. And in fact, naloxone is a drug that reverses the effects instantly of an opioid overdose. So it's an important drug we're going to talk quite a bit about. No? Okay. There are different varieties of receptors. Ionotropic and metabotropic or G-protein coupled. The ionotropic receptors are much faster. They respond very quickly because they open ion channels in the cellular membrane, which is why they respond so quickly and why they cause the cell to respond so quickly. They're called ionotropic because, again, they cause those ion channels to open. As soon as a neurotransmitter binds with them, the ion channel opens or closes and causes a very quick response in the neuron. So this is a direct reaction to the neurotransmitter, and it causes a direct either inhibition of an action potential or the creation of an action potential. So they're contrasted with what were called metabotropic receptors. These respond much more slowly. They respond indirectly, and they're called metabotropic or G-protein coupled receptors. And essentially what this does is it initiates a second signal or messenger inside the neuron. So when a neurotransmitter binds with a metabotropic or G-protein coupled receptor, it causes a change in this protein called an isomer, which then causes a G protein, which then causes another reaction within the cell. So it stimulates the synthesis of a secondary messenger or binds to an ion channel and causes it to open, but it causes a secondary action. It doesn't directly affect the cellular process. So these metabotropic or G protein coupled receptors are slower and have a very different kind of response profile. Now once synaptic transmission has occurred, it has to be inactivated, that it has to be terminated so it's not constantly firing. So in order to have a system that creates information, you have to have some kind of system for starting and stopping neurotransmission. The first part of this is metabolism. The second is reuptake. And the third is reuptake by a glial cell. Metabolism occurs by which the neurotransmitters are metabolized in the synapse itself by different enzymes, things like acetylcholinesterase or other monoamine oxidases that convert those neurotransmitters into other substances. Reuptake occurs where the neurotransmitter is taken back into the cell, and of course autoreception just simply stops the process of initiating any more release of neurotransmitters. Glial cells will uptake glutamate, which can cause some issues we'll talk about later on. So the major events that occur at a synapse, we have synthesis of smaller neurotransmitters such as acetylcholine. Larger neurotransmitters are usually created in the cell body or the soma. An action potential causes calcium to enter the cell, releasing the neurotransmitter, which causes the vesicles to bind and release a neurotransmitter. The neurotransmitter binds to the receptors, gets separation from the receptors. We get reuptake of neurotransmitters by the transporter proteins. Postsynaptic cell releases retrograde transmitters that can further release from the presynaptic cell, or further release from the presynaptic cell. So, for example, endogenous endocannabinoids function in this way. They are released by the postsynaptic neuron to actually cease neurotransmission from the presynaptic neuron. So all of these are major events that occur at a synapse. So again, here is a nice full summary of this whole process. Wouldn't expect you to know the nitty-gritty details, but this is an important part of understanding how neurotransmission works and understanding how drugs can affect this process. So for the rest of this lecture, I want to spend the time talking about specific neurotransmitters and the neurotransmitter systems within the brain and other parts of the nervous systems that are influenced by these neurotransmitter systems. So you can break up neurotransmitters into different categories. Most often they are categorized either by their function or by their structure. So for the most part, by their structure. So that we have the amines. We have quaternary amines, which is acetylcholine, and monoamines, which include two different classes of catecholamines and indolamines. The catecholamines are norepinephrine, epinephrine, and dopamine. The indolamines are various kinds of serotonin, which include various versions of 5-HT. We'll talk about how there are versions of serotonin and versions of dopamine. So there are currently at least 4 different dopamine receptors and dopamine neurotransmitters. Similarly, there are several different versions of serotonin. Amino acids include gamma-aminobutyric acid, which is a very important inhibitory neurotransmitter, glutamate, and glycine. The neuropeptides include beta-endorphin, dynorphin A, and other opioid peptides. Other peptides include oxytocin, which is an important binding hormone, substance P, which is involved in pain, vasopressin, which is involved in blood pressure, hypothalamic releasing hormones, and then gases like nitric oxide and carbon monoxide can also have effects at different receptor subtypes. Acetylcholine is one of the most common and most important neurotransmitters. It's first to be recognized because its peripheral actions, that is, it has effects at a lot of peripheral sites, particularly within the muscular system, things like respiratory, cardiac systems. All of those involve acetylcholine. We call these receptors cholinergic receptors. These include 2 different kinds, nicotinic and muscarinic receptors. Acetylcholine esterase is an important enzyme that breaks down acetylcholine in the synapse. A variety of drugs have targeted acetylcholine esterase. They're called ACE inhibitors, which inhibit the functioning of this particular enzyme in order to increase the amount of acetylcholine and hopefully improve memory. These drugs are usually used to treat things like dementia, and that's what ACE inhibitors are for. There are reversible ACE inhibitors and irreversible ACE inhibitors. Irreversible ACE inhibitors are generally toxic. These include things like pesticides and nerve gases. They kill by inhibiting acetylcholine esterase, so a huge buildup of acetylcholine, which causes massive malfunction of nervous systems, and as a result, death. Reversible ACE inhibitors are often considered cognitive enhancers, and they're used for doing things like treating Alzheimer's, which I was talking about a moment ago. So ACE inhibitors are particularly important. You've probably heard in the news recently the nicotinic pesticides have been implicated in beehive colony collapse. And so a lot of the nicotinic neonicotinoid pesticides are being banned in a variety of locations. And so keep in mind, not all effects on neurotransmitters are going to be positive ones. And so we can have some very negative effects on neurotransmitters by affecting those systems. The cholinergic system has projections from a variety of places in the brain, particularly coming from the basal forebrain up through all of the cortex, particularly motor areas, the hippocampus, cerebellum, areas involved in breathing, autonomic nervous system, etc. We'll talk about anticholinergic drugs. and their influences, particularly, they can cause things like amnesia. Scopolamine is a well-known drug that can cause amnesia, but is also useful for treating motion sickness, and so it has a direct effect on those systems. The nicotinic receptors in the cholinergic system control the skeletal muscles, whereas the muscarinic receptors tend to control involuntary muscles, things like heart, breathing, that sort of thing. So anything that targets the muscarinic receptors are going to influence involuntary muscles, the nicotinic receptors, those skeletal muscles. The catecholamines are synthesized from tyrosine, which include dopamine, norepinephrine, and epinephrine. So it's simply a transformation of tyrosine into various other organic molecules like dopamine and norepinephrine. Their termination occurs through reuptake and through an amino acid, monoamine oxidase, which is an enzyme which breaks down the catecholamines. A number of antidepressants are monoamine oxidase inhibitors, or MAOIs, and we'll talk a great deal about those when we get to antidepressants. Norepinephrine is an incredibly important neurotransmitter. There are axonal projections that travel down the spinal cord where they can exert an analgesic action, that is, they can inhibit pain. So when we have a sympathetic nervous system response, norepinephrine is the primary neurotransmitter that is released. And the reason why norepinephrine inhibits pain is during those high stress times, pain is a distraction from survival. And so this is one of the reasons why athletes that have been injured don't know how injured they are, because they have a great deal of norepinephrine going, and so they don't realize how much pain they might be in. So the release of norepinephrine produces an alerting, focusing, orienting response. So people with a high degree of norepinephrine are highly focused, highly alert, sometimes even paranoid. Norepinephrine is also, however, associated with positive feelings of reward, as is dopamine, we'll talk about in a moment. Dopamine and norepinephrine often co-occur in drugs of abuse like cocaine and amphetamines. Both neurotransmitters are involved in those particular drugs and have important implications for their behavioral effects. So if we look at the dopamine pathways, the hypothalamus to pituitary gland dopaminergic pathway controls a variety of hormones. So this is going to release various hormones that influence things like eating, sex drive, that sort of thing. From the substantia nigra to the basal ganglia, this dopamine pathway is involved in motor coordination. This particular pathway is implicated in Parkinson's disease. And then finally we have the ventral tegmental area, which projects to the cortex in the limbic system, which is pictured here. This is part of the brain's reward pathway, and this reward pathway is going to be very important. We'll be spending some time talking about that when we get to the neurobiology of addiction. The reward pathway is a particularly important part of that, but that mesolimbic pathway and mesostriatal pathway and mesocortical pathway are all part of dopamine systems. The mesolimbocortical pathway is part of the reward system. The mesostriatal pathway is part of the motor coordination system. So dopamine is particularly important. Serotonin pathways. Serotonin is synthesized from tryptophan. This is one of the reasons why there's this sort of legend about tryptophan and Thanksgiving turkey making you very sleepy. It's actually the heavy load of carbohydrates which tends to make you sleepy. But tryptophan is an important protein which can be synthesized into 5-HT. There are 7 different 5-HT receptors with 6 different subtypes. So pretty big serotonin system. These are ionotropic receptors. 5-HT3 seems to be one of the most important of those. We'll talk about that later on. There are a variety of serotonergic pathways. They largely parallel the dopamine pathways. So if you look at the mesencephalic serotonergic cells, project to very similar areas. There are, of course, exceptions to that. We have projections from the Rafe nuclei to the cerebellum, which is from the brainstem to the cerebellum. We also have various other subcortical projections, but very similar. to the thalamus, hypothalamus, basal ganglion cortex that parallel the dopamine pathway. So oftentimes these two go hand in hand as well. Glutamate is another important neurotransmitter that is involved in a variety of processes. This is the major excitatory neurotransmitter in the brain. It comes from the metabolic pathway, so this comes right out of that Krebs cycle that we all learned so much about in biology. Or it can come from glutamine via glutaminase. Binds to several different receptor types. The NMDA receptor, which is the N-methyldeaspartate receptor, which is very important, which we'll come to talk about a great deal. It's also another nicotinic receptor. The kainate and the AMPA receptor types. The NMDA is mediated by glutamate and glycine serine. NMDA requires membrane depolarization by one of the other neurotransmitters. And the NMDA receptor is certainly involved in memory formation. And we'll talk a great deal about how the inhibitory neurotransmitter GABA is involved in memory formation on that NMDA receptor. For some other notes about glutamate, there is some belief that epilepsy is related to glutamate. So when we start talking about drugs that are used to treat epilepsy, understand that glutamate is some of its ultimate target. There aren't a lot of drugs that we will talk about that have major effects on glutamate, except for PCP and ketamine, which are psychedelic drugs, which are glutamate antagonists, so they actually reduce the functioning of glutamate. We'll talk more about those when we get to psychedelics. So those are also very important drugs. Important to stop for a second and talk about the NMDA receptor. The NMDA receptor is particularly important for psychology. It has some very unusual characteristics and has a lot of binding sites for drugs that we're going to talk about. So in addition to glutamate, another amino acid, either glycine or serine, needs to be present on the receptor for activation. They don't need to be stimulated by glutamate, but they also need to be sufficiently stimulated electrically. So there is an electrical component and a need for glutamate. And so you get a variety of receptor sites on the NMDA receptor, which various neurotransmitters can bind to, in addition to them responding to the electrical activity of these the cell to get this transmitter to actually function. So this NMDA receptor is particularly important, and it has a variety of binding sites for various neurotransmitters. So they play a role in regulating synaptic plasticity. So trying to establish new synaptic pathways is essentially how we learn. And so anything that involves the NMDA receptor is probably going to involve memory and certainly learning. GABA, or gamma-aminobutyric acid, is a universally inhibitory neurotransmitter that is found in high concentrations in the brain and the spinal cord. We'll talk a great deal about GABA. GABA seems to be implicated in the effects of alcohol, and it's certainly affected by a variety of benzodiazepine sedatives like Xanax, Ativan, Valium. These are all GABA agonists. That is, they increase the functioning of GABA and as a result are very inhibitory in nature. GABA is made from glutamate under control of the enzyme glutamic acid decarboxylase, or GAD. It's metabolized by GABA transaminase. And the receptors are both GABA A and GABA B. Transmission of GABA is usually by reuptake with transporters on the neuron or glial cell. Again, GABA is an important inhibitory neurotransmitter. We will talk a great deal about how amnesia can be caused by drugs that induce the functioning of GABA, and also how when people quit smoking, that can cause changes in the regulation of GABA, and as a result, people's memories don't function as well following their attempts to quit smoking. So we'll talk more about that. Again, benzodiazepine sedatives are strong GABA agonists and universally have negative effects on memory. Some have very strong effects on memory. Drugs like midazolam, which is Versed, actually causes very, very dense amnesia. So those will come up again when we get to talking about sedative hypnotics. The peptides, we will briefly mention now. They're small proteins or chains of amino acid molecules. They're just attached in a specific order. These peptides generally are mostly involved in opioid receptors. The mu, delta, and kappa are all involved in modulation of pain. We'll also talk about endorphins and enkephalines and substance P. All of these are involved in maintenance or function of all of the opioids like morphine, heroin, etc. And so we're going to talk a great deal about these receptors when we get to pain perception. So to wrap up today's discussion of brain and neurotransmitter, just take a look at the review of these different neurotransmitter synthesis. Acetylcholine is metabolized from various parts of the diet, acetyl coenzyme A and choline. Phenylalanine becomes tyrosine, can become dopa, then becomes dopamine, which becomes norepinephrine, which can become epinephrine, and then tryptophan becomes serotonin eventually. We'll also talk briefly about steroid hormones and how they have very similar metabolic pathways. But for now, that is it for our discussion of brain structure and function. For our next lecture, we will start talking about pharmacokinetics.